Hello students, in today's class we will be studying about the next type of sterilization, that is sterilization by filtration. Now what is this sterilization by filtration? You all know sterilization is a process wherein you are getting rid of the microorganisms. Now here you are not killing the microorganisms, but filtration is a process wherein the solid particles which is suspended in liquid or probably gaseous fluid or in gas or in air are removed using a filter pad or a filter medium. This is called as filtration. Filtration, basically there will be a filter medium or a filter pad of a specific pore size and of specific thickness. Now I want to filter out something, that is some liquid or some gaseous material which is passed through this filter medium wherein the solid particles get separated out. So that is called as filtration. As you see here in this picture, there is a solution which contains solid materials as well as the liquid material. This is the filter pad. The solution is passed through the filter pad of one particular pore size. Only the liquid portion gets collected, which is called as a filtrate, and the solid particles get attached, gets absorbed or gets collected on the filter pad. Who is the size of this material will be bigger than the pore size. Now here solid particle means to say here the microorganisms who gets separated out. Hence when I pass the solution which contains microorganisms, when I pass it through a filter pad, the microorganisms gets collected here on the filter pad and I get a solution, that is the filtrate which is free of microorganisms. Now why do I need to have, or why do I need to follow this filtration process for sterilization? Is it required? Yes. Now generally heat is applied for sterilization purpose to get rid of the microorganisms. But what happens is there could be certain media or certain media components which can or which could be sensitive to heat. Now in such cases the media component or the media, if it is sensitive to heat, then I need to go for alternative methods. Then I use this filtration as one of the methods here. Which are those some of the ingredients? Like example, if I am using blood or I am using serum, egg yolk, etc. Now these are the components when added to a medium. I mean these are the some of the ingredients when I will be adding to the medium. Now these components definitely cannot withstand high temperature. In such cases I will use filtration as a process of sterilization. Now moving on to the different types. What are the different types of filtration? Laminar airflow method, Seeds filter, or I mean to say what are the different filters that are available? Laminar airflow, Seeds filter or asbestos filter, Porcelain filter or Chamberlain filter, diatomaceous earth filter or Bergfeld or Mandler filter, membrane filter or ultra filters. Now these are the different types of filters used according to the requirement or according to the pore size or according to the microorganisms that I need to separate out. The first one is LAF. Now what is LAF? LAF is laminar airflow bench. It is laminar airflow bench. Now this is an instrument which will sterilize the air or it will sterilize the filter. Now if you see in all these types, laminar airflow, LAF is a method which is used to sterilize the air or filter out the air, whereas all the other four types is a method or are filters which is used to filter out or sterilize liquid medium. So as I told that this is a instrument which is used to sterilize or filter out the air. And this is generally used for inoculation purposes. If I want to transfer any medium, or if I want to pour a medium, I want to transfer an organism, do inoculation, all such instances I will use this laminar airflow bench, which is a closed cabinet here. And there will be a glass door. It has switches here, 3 switches, one for the light, another one for the UV lamp, and the third one is for the blower. Now there are definitely, when we talk about filters, filtration process, filter pads should be available. So what is the filter used here? It is called as HEPA filters, HEPA. That is high efficiency particulate arresters or high efficiency particular air filters. Now these filters, if you see the construction of the laminar airflow bench, this is a closed cabinet here with a glass door. The glass shutter can be opened when we are working. A person will be sitting in front of that working bench. This is the working bench and there will be a gas burner switched on there. Now before starting the experiment, what I need to do is I need to clean the tabletop and then close the shutter and switch on the UV light for 20 minutes, because by the UV rays the microorganisms can be killed in the working platform. Next, what I need to do is I need to switch off the UV and switch on the blower, and then open the shutter, switch on the burner and do my inoculation or any work in front of the burner. Now, what is the working principle? Where are these HEPA filters located or placed? HEPA filters could be located here on the top or at the back side here. Now, if you see in this diagram, the HEPA filters are fixed here. How is the working principle? It's basically working on suction force. Now, there will be atmospheric air outside. So, what happens here is the laminar airflow bench, when we switch on the blower, it sucks in the atmospheric air, and then the blower will push the air that is sucked inside the laminar airflow bench and it will push through the HEPA filters. This is the filter. It will push through the filter and only the sterile air will be pushed out of the filter, and this is a working platform and this will be filled with sterile air, and this is a continuous process until I switch off the burner. Now, what is the efficiency of these HEPA filter? It can remove around 99.97% of the air that is coming out is 99.97% sterile. And what is the particle size? What is the size, pore size of these HEPA filters? It is 0.3 micrometer. The pore size. Now, as I told you, this is an instrument which is used to sterilize the air, to filter out the air. Advantages of this is that no other instrument can be used for sterilization of the air, but this I will get sterile air. Disadvantage, a sophisticated instrument which is very expensive should be used here. Moving on to the next kind of filter that we had listed out is the seeds filter or asbestos filter. Now, when we talk about seeds or asbestos, what is asbestos? These are the asbestos sheets or asbestos fiber which we generally see when during construction the rooftop asbestos are used. So, what is the thickness of these asbestos filter? Around 2 to 6 mm thickness. Such should be the thickness, and the pore size varies from 0.01 mm to 5 microns. Okay, so this is the 0.01 to 5 mm. This is the pore size of the asbestos filter. This is how the asbestos filter or seeds filter look like. This is the place wherein the sample is added, and here the filter pad is placed, that is the asbestos filter, and through this the sample filtrate moves in and it gets collected in a beaker or a conical flask. There are certain precautions wherein I need to take care, that is, we need to handle this entire setup carefully. The pore size I need to select based on what kind of organism or what is the sample that I need to sterilize. And if the filter is handled in a harsh way, it may lead to the damage of the filter. Now, this is the skeleton part of the asbestos filter. Here is the place wherein I add the sample. This is the filter pad, and this is through which the filtrate gets trickled down. This cannot be, it's a very sensitive product, it cannot be mishandled. And the filters has to be used only once. You cannot reuse the filter pads here. Moving on to the next one, that's the porcelain or Chamberlain filter. Now porcelain is basically made up of a mixture of components, that is aluminium silicate, iron, calcium, magnesium and diatomaceous clay. All these components are mixed together and subjected to a very high temperature around 800 degrees centigrade, and then when you do this, these components gets melted and then you have to cool them. When you do this, a pore size of 0.65 to 15 mm is reached. Now this is the pore size, and then you get the you will be able to filter out the microorganisms or filter out the unwanted materials. Now how does it look? Now this is how the filter looks like. This is the filter, this is the porcelain filter. This is the place, the porcelain filter is fitted to a long glass tube and also a collecting chamber, maybe a round bottom flask or a conical flask. Now this is the filter. Whatever sample you need to filter out is added here in this long glass jar. Now the sample from here moves through this filter into the mantle. Now this is called as the mantle. It gets collected here through a process called as diffusion. You all know what is the process of diffusion, wherein the solvent molecules moves from the region of its higher concentration to the region of its lower concentration through a semi-permeable membrane. So this filter acts as a semi-permeable membrane, and then the sample gets collected here. This is the filter. Now this can be reused. The filter can be reused here. Moving on to the next one is your Bergfield, Mandler or diatomaceous earth filter. Now here the filter pad or the filter is made up of diatomaceous earth. Now here, how is it prepared? The diatomaceous earth material and the asbestos sheets or fibers are mixed together in uniform proportion and is subjected to very high temperature around 1000 to 2000 degrees centigrade and at a very high pressure. Now when this is subjected to very high pressure, what happens? The components melt down, diatomaceous earth melts, and then immediately it is cooled down. When this is cooled down, the porosity that you achieve after the cooling down is 0.3 to 0.6 mm. So this is the filter that you get. This is a diatomaceous earth filter or Bergfield filter. This is called as a mantle. That's why it is called as a Mandler filter also. Now here, whatever I want to separate out is filled in this place in this jar, and then again through the process of diffusion the sample moves. This acts like a semi-permeable membrane. This mantle acts as a semi-permeable membrane, and the sample moves into it through the mantle and it gets collected here, and slowly the sample, the filtrate, the pure filtrate gets trickled down and it is collected in the conical flask or the beaker. So this is how the sample is filtered. Now when is this filter used? Generally this filter is used to filter out water, or also sometimes if you find these in industries, like where there are lumps formed in for example in juice, it has to be a flowy component, but then suppose if there are lumps, or probably in food or medicine or in paints etc., it has to be a flowy component, but if lumps are found, it is generally used to separate out these lumps. And also of course microorganisms. Now this filter also is used to separate out bacteria, and the disadvantages of this is that it is a slow process, and then it has to be there are chances that this mantle can be blocked, and frequently you need to clear this. Moving on to the last filter, that is the membrane filter. It is also called as a molecular filter. It is also called as millipore filter. The reason being, it was first introduced in a company called as Millipore Filter Corporation, which is in USA, and that's the reason it is called as a millipore filter. It can separate out all the microorganisms, that is even viruses could be separated out using this membrane filter, and generally this is used in water purification. This is how the setup is. Here this is the place wherein the sample is added. Here membrane filter is placed. This is the membrane filter. If you see the skeletal structure, this is the place where sample is poured. Here the membrane filter is added, and here the filtrate is collected. The pure sample is collected. Now every time, and this entire setup is connected to a vacuum. And hence, since it is connected to a vacuum, there is a suction force. When compared to all other samples or all other kinds of filters, this acts fast, or the process happens faster because it happens through suction. Now, when I add the sample here, the sample is added in a sample collector, and it moves through the molecular filter or the membrane filter. The microorganisms get separated here. What is the pore size? The pore size is around 150 micrometer, and then it gets collected. All the microorganisms get separated here, and the pure filtrate is collected here. Now, what are the advantages and disadvantages? Advantages is a very quick process. Since it is happening through suction, it doesn't take time for the sample to trickle down. It just pulls, it just sucks. Also, it's very easy. Even if there is blocking, I can simply dispose this membrane filter and I can use a fresh one. And it is very easy to remove the membrane filter and add a fresh one. Also, I can also make out, or I can find out the quality of the sample here, or what kind of organisms were present. That is, if this filter pad is taken, and it is since the microorganisms are collected here, I have to simply remove this and place it on to a suitable medium onto a plate, and then incubate it, and next day after 24 hours check for the growth of the colonies. If there are microorganisms, definitely the colonies will come out. So not only filtration, I can also check for the quality or kind of microorganisms that were present in this particular sample. It is generally used in water purification now. So these are the different kinds of filters. Now the advantages of this is, those materials which are heat sensitive can be can be sterilized by filtration. What are the disadvantages? Disadvantages is that only air and the liquid form can be sterilized here. If it is a solid media, if it is some component which is sensitive to heat, and if it is a solid substance, then definitely I cannot use this method for sterilization. Thank you.